Bismillah ar-Rahman ar-Rahim. Today we're going to discuss a few practical skills in handling paper 3, which is going to be in the November exam as well as in the next March and the June exam. So just giving you a little clue on how to handle the paper 3. So I wish all of you the best of luck in these exams, and I hope this video is helpful. Thank you very much for watching. As everybody knows that this paper is for 2 hours, and you must be very careful in trying to finish the entire paper in the time that is allotted to you. You know that this is all this which is written, you know that, and you know you do not write in the barcode. You may use an HB pencil, write in dark blue or black pen. Answer all the questions. Now, before you proceed, read carefully through the whole of question 1 and question 2. Plan the use of the 2 hours to make sure that you finish the whole of question 1 and question 2. Enzyme E catalyzes the hydrolysis of starch to maltose. The progress of this enzyme catalyzed reaction can be followed by measuring the time taken for the substrate, which is starch, to disappear. You will investigate the effect of an inhibitor C on the hydrolysis of starch. You are provided with the material shown in table 1.1. You must look at it as you're doing this in the lab. E is the 1% amylase solution. It's an irritant, and the volume given to you is 10 cm cube. Then the risk. Then S is a 1% starch, none, 50. And 2% copper sulfate, harmful irritant at 20 cm cube. You see, these volumes are very important because you should be able to use most of this reagent. So you must know when you have to repeat these experiments, and that of course would be given a mark, as we can see as we read on the question. W is distilled water, iodine solution, and U is the unknown concentration of copper sulfate solution. And this is harmful, and there's 10 cm cube of that. Then it says if any solution comes in contact, wash off immediately under cold water. It is recommended that you wear suitable eye protection. You're always given that. You're supposed to wear the goggles, but most of you don't wear them. Think about the hazards of using the material in table 1.1. Decide whether the risk of using E, S, and C is low, medium, or high. So you have to only give me for E, S, and C. Do not give me for the others. So as you all know, that the risk for E is medium or high, for S is low, and for C is medium or high. So E is medium, or even if you had said high, be on the safe side, you can write medium. And S, of course, was what, 1% starch solution. You know, starch is nothing, so it's low. We all have starch in our bread and our roti and the rice. And then copper sulfate is, of course, as usual, it's also medium. So either you said medium or you said high. I'm not saying you could have said this. Here also you could have said medium or high. Now, this was only for it's only for 1 mark. But be careful that you don't lose this mark. So it's usually low or medium, and of course anything which is like starch would be low. The others could be medium or high. That is the usual pattern that we follow. Then the next part says you will need to make a serial dilution of the 2.0% copper sulfate solution C, which reduces the concentration by a factor of 10 between each successive dilutions. Figure 1.1 shows you the first 2 beakers you will use to make your serial dilution. Decide which other concentrations of copper sulfate you will use to make your serial dilution. Complete figure 1.1 by drawing as many extra beakers as you need for your serial dilution. For each beaker, state under the beaker the volume and concentration of copper sulfate available for use in the investigation. Use one arrow with a label above the beaker to show the volume and concentration of copper sulfate added to prepare the concentration, and use another arrow with a label above the beaker to show the volume of W added to prepare the concentration. So 3 things that you have to do. Read this very carefully and understand that. I'm sure 2 years you've done loads and loads of such practicals and you're all very expert at this now. But some of you, of course, are not very expert, so just helping you all out. Now this is the figure 1.1, and you can see this here. They have said you've added, look at it very carefully, they've said you've added 10 centimeter of 2% copper sulfate solution and there's 0 water in it. So originally was 10 cm cube, and then this is the 2% copper sulfate, and you have to dilute it by a figure of 10. So 2 over 10 would be what? 2 over 10 would be is equal to 0.2. So this beaker would be 0.2%. So you see this one was 2% copper sulfate. Then we have 0.2% copper sulfate. Then further 0.02%. So from 2 it went down to 0.2%, then 0.02%, then it will be 0.002%, and then 0.0002%. So it's been diluted by, so we've got now 1, 2, 3, 4, 5. And that's what I always advise, that you must have a total of 5 beakers. That is what is preferable in all the situations that are given to you. So 0.2, because we are dividing it by 10, because it said in the question, here it said in the question, if you look at the question, it said here factor of 10, between successive dilutions. So the factor of 10 becomes from 2%, it becomes, so this will 2% copper. So then this will be, of course, now I'll complete the table for you and then you can see how I'm going to do it. So if you can see here 10 and then 9 centimeters, so that means 1 centimeter of what? Of the 2% copper sulfate this is the one. So you've added 1 cm of this here, and then you will add 2%, this will be the 2% copper sulfate. And so you're left with 9 cm back. This gives you the clue. So you've taken 1 centimeter here, and then now what are you going to do? You're going to add 9 cm cube. You're going to add 9 cm cube. 9 cm cube of water. So this will become 10, and then when you take out another 1 cm cube, now you'll take out another 1 cm cube here, and then you'll put it in another beaker. I'm going to rub this off so that then I can draw all the beakers here and show them to you in the correct manner, but I'm just showing it to you. So this would go here, you'd get this into another beaker here, and then to it we will add another 9 cm cube of water, and then again you will take 1 cm cube out of it into another beaker, and then add 9 cm cube of water again to this. I'm using different colors so that you can understand it. So this is 4, and then you will take another one out, and then you will add it to another 5th beaker. So now this will be 0.2%, this one would be 0.02%, this one would be 0.00002%, and this one would be 0.0002%. And of course in the last one you'll be left with 10 cm to be used. In the others you'll have 9 cm that will be used. The next one is now you take out of this and then you put it in here. And then you have to write on this one, what is this one? This is 1 cm cube of 0.2, because this is 0.2, 0.2% C. And then you add to this also 9 cm water. Right? But then this solution would be what? Now this one would be 0.02%. And this will be 9 cm cube of this. Why? Because we're going to take one out. So this will be 9 cm cube of solution C to use, because we have to write this C to use. And then you take again, serial means you keep on doing this again and again. So you've taken again 1 cm cube of 0.02%. This was 0.02%. And then you added again 9 cm cube of water. 9 cm cube of water. And now this would be 0.002% of C to use. Of course, I'm using different markers, but you can have to use one pencil, so you need to write it very carefully and do this. You can't lose the marks of this. This is the easiest part of the practical. So the last table is going to be, you'd actually have 10 cm here to use, because now you're not taking it anything out of it. Or in the others, you all have, this is 9 cm, and again here this is 9 cm, and then this would also be 9 cm cube. So I'm sorry I can't get it all to see it all in one go, but let's have a look at it. Maybe you can just pause the video here and have a look at it. And this is what I expect you to understand. Once you have 2%, you put that here, and then you put a 9 cm cube of water in it, and then you added 1 cm, then you took out the 1 cm cube. I've drawn two different colors to make you understand, and you added water. Every time you added the same water, and every time you take out the same, so then you are diluting it by 10 times, because actually you had 10 cm cube here, so you've taken one out and added 9 water to it, so it has been diluted 10 times. So this is how you would have done this, like this, and you get these 5 beakers, and you would have all these 5 beakers here, and this is for 3 marks. The 3 marks are for all these, for taking 1 cm out every time, adding 9 cm of water, and then getting these concentrations correct: 0.2, 0.02, 0.002, and 0.0002. So if you got all that right, you got your 3 marks. Then as we continue with the question, it says carry out step 1 to step 19. Prepare the concentration of copper sulfate as decided in A2, which we've just done. Use a glass rod to mix the copper sulfate solutions and water. Label the test tubes with the concentration of copper sulfate solution prepared in step 1. So you've got all the test tube, you label them, and you have 5 of these, so you label them 2%, 0.02, and so on and so forth. Now label another test tube W. So you label another test tube W, wipe the tile clean with a damp paper towel, and then dry the white tile. So you first clean it with a wet cloth and then you dry it. And then label the white tile as shown in figure 1.2, the numbers indicating the sampling time in seconds. So this is 15, then 30. So label the white tile as shown, the numbers 15, 30, 45, 60, 75, 90, 105, 120, 135, 150, 165, 180 seconds. Put one drop of iodine. So what is the color of iodine? Color of iodine is brown. So you put one drop of iodine here, one drop of iodine here, and you keep on putting this. Put one drop of iodine on the white tile at each sampling time as shown in figure 1.2. Put 1 centimeter of W in the test tube labeled W. So you put 1 centimeter of W here. Please remember centimeter cube and ml are the same thing. Put then 3 centimeter cube of S. And then we have 3 centimeter cube of S into the test tube. You mix well, remember S was starch. And then you put 0.5 cm cube of E. And then you put 0.5 cm cube of E. And E was the enzyme, as it told you in the beginning. What was E? E was the 1% amylase solution. S was the starch and C is the copper sulfate. So then you put 0.5 cm of E into the test tube, use a glass rod to mix. Start timing. After 15 seconds, use the glass rod to transfer a drop of the mixture from the test tube onto the iodine that is labeled 15 on the white tile. Then clean the glass rod with a paper towel. And then repeat step 11 to step 12 at intervals of 15 seconds until the iodine does not change color. You see, because when you add starch, it becomes blue-black. But if all the starch is finished, then it won't change color, it will remain brown. So record the time taken for the starch to disappear in an appropriate table in A3. If the starch has not disappeared in 180 seconds, record more than 180. We don't want you to keep on waiting and waiting, or you'll waste your time. Repeat steps 4 to 14, replacing 1 centimeter of W with 1 centimeter of the lowest concentration of copper sulfate. Which one was that? 0.0002, the lowest concentration. Then repeat step 15 with the other concentrations of copper sulfate. Record your results in appropriate table. So first it was W, and then it was this one. Read the question very carefully, because in the table they're going to give you a mark for that. If you didn't follow this instruction, well, you're going to lose a mark then. Now, as you can see, this is 5 marks, and this is a 5 marks which I check a lot of papers and I see a large number of students, majority of students getting these 5 out of 5 marks. And they're actually for just recording it, and there's a rule by which we do this. I'm just going to explain that to you. Please, everybody, remember that. Now, as you can see, I have just cooked up the results. Now, theoretically, when the copper sulfate, which is an inhibitor, will increase, then it will take more time. Why would it take more time? Because more the inhibitor means more the inhibition. So it will take more time for the starch to be hydrolyzed to maltose. Now, what are the marks for? The marks is for giving me this heading. The fact that you gave me this heading gets you 1 mark. The fact that you gave me this heading gets you a 2nd mark. And then you gave me readings for all the samples, and you did not write seconds here. If you wrote seconds in the body of the table, well, you lose a mark. Or if you wrote percentage here, because the percentage is given in the heading. If you wrote percentage here, you again lose a mark. So please do not write the units in the body of the table. Do not write the units. Do not write second here, second here. This would be the biggest mistake of your life if you did this, because that loses as a -1. Or if you wrote percentage here, this again you lose a mark for that as well. You're not supposed to write the units. You're supposed to only write the units here, and the units here, like seconds is written here, and that's it. Now, the 5 marks are one for the heading. For this heading, this gets you 1 mark. Then this gets you a 2nd mark. A 3rd mark is the readings for all the samples. The 4th mark is for the correct trend. Theoretically, time for iodine to remain yellow increases with increasing concentration. So it would increase, and you wrote the results to the nearest whole second. You did not write 75.10 seconds or something like that. Please do not write. Seconds are always to be written in whole seconds, never as a fraction. So that got you your 5 out of 5. Then it says 17, label a test tube with the letter U. Repeat step 4 to 13 with U instead of W. Record in A4 your results for U, state the results for U. Well, the result for U was, say it was this, 150 seconds, right? Anything, it could be anything. 150 seconds. And you correctly estimate, so the correct estimate would be this. Would be what? The copper sulfate would be 0.2%. So this could be anything. You could write anything, it will all be correct. Just had to record the result for you, and second is not something that what was the correct estimate, but whatever was the estimate from your reading. Your reading was, it was 150 seconds. So if 150 seconds is 0.2%. So remember that, and everybody's answer could be different. It's not necessary that the answer of all the classes got to be the same. Cambridge gives you this opportunity to get that mark. So you had 1 mark for this and 1 mark for this, because you estimated it correctly. If you said 165 seconds, then you know it would have been 2%. Or something else, if you had said 120, then it was 0.02%. So you just have to do it from your table. And as I always say, sometimes you need not even do it and you can just write it. Then coming to part 4, state one significant source of error in this investigation. Explain why this is a source of error. Yeah, well, difficulty in judging the exact time of color change. You're doing it after 15 seconds. What if after 5 seconds things have changed? Color may change between the time intervals. And then using a glass rod causes drop sizes to be an unequal size. So source of error, difficulty in judging the exact time of color change. We are doing it after every 15 seconds. And then the color may change between the time intervals. Maybe between 15 and 30 at 18 seconds it has changed color, but you're going to sample it after 30 seconds. And then the glass rod drop size could be of unequal size. So difficulty in judging the color change, then color may change in between the time interval, and glass rod drop is of unequal size. Then coming to part 7, suggest how you could make one improvement to the independent variable so that a more accurate estimate of the concentration of copper sulfate in U can be obtained. As a very simple one comes in a lot of papers, more concentrations between the two concentrations that lie each side of the estimate. Like for instance, if you said the estimate was 0.2%, so then you would need to do more, just less than zero point 0.2 and a little more than. So you could say 0.2, 0.1 and 0.3 or something like that. So you would have to have more concentration between the two concentrations that lie on each side of the estimate. So more concentrations between the two concentrations that lie at each side of the estimate. Like your estimate was say zero point 0.2. So then zero point 0.3 and zero point 0.1. And you'll be more sure that it is really 0.3 or zero point 0.1 because you have 0.2 and then you have 2%, or you have 0.2 and then you have 0.02. So you have to remember this is that you'd have to see whether it is just this or a little more than this or a little less than this. Then part 8 suggests how you could make two improvements to the measuring of the dependent variable so that a more accurate estimate of the concentration of copper sulfate in U can be obtained. Use colorimeter, standard colors to compare more intermediate times, choose endpoint that is dark brown, use pipette for iodine or sample. You see, use a colorimeter. That would be, of course, a very inaccurate method. The standard colors to compare. If you had made a color standard with known concentrations of starch, and then which it doesn't have, then you would exactly know how much was that, or the copper inhibitor story. More intermediate times. Choose endpoint that is dark brown, and use a pipette for iodine or sample. Using a glass rod was not a very good idea because that would change the volume of the sample or change the volume of the iodine. Then of course we come to the next part. A student carried out an investigation on the effect of temperature on the activity of enzyme E. The results are shown in table 1.2. Now remember, the first column is always the independent variable. The first column is always the independent variable, and this is always plotted on the x-axis. That's why I make this mnemonic of FIX. So this will always go on the x-axis, and this is the rate of reaction in arbitrary units must go on the y-axis. This is a rule. And you must write the whole thing you must write, and you must write the whole of this, whatever is given. Do not abbreviate, please. And then think of the scale, because you have to fit it from 5 to 49.5. And the lowest here is 57 and 102.5. So you've got to fit the entire grid. So be careful that you fit this hole and also lengthwise the whole of it. As you can all see, the graph is for 4 marks. Now if you just did this, which means you just labeled the axis, temperature, degrees Celsius, and rate of reaction, arbitrary units, well, you get 1 mark for that. And it's sad when I'm checking papers and I see somebody who's not done that. They've not labeled the axis. And I feel like, that's so irresponsible and so casual of the student of doing such a thing. I mean, your parents pay a phenomenal amount of money for you to take the exam, and you casually take the exam and you don't label the axis. It makes me very sad. Really makes me very, very sad. Then you number it. Every 2 centimeter you write. So the scale that was expected of you was 10 degrees Celsius to 2 centimeter, and labeled every 2 centimeter. You see, this is 2 centimeter. Remember this. So this is 2 centimeters. So you label it every 2 centimeter, and this would be, of course, this would be the 0 here, and 10, 20, 30, 40, and 50 in the end. Then the y-axis. Now, as you can see here, this was the highest figure, which is 245, and the lowest figure was 57.5. So you could have started also from 50, but of course you started from 0, 50, 100, 150, 200, and 250, and you labeled this on the y-axis, and you got, so the correct scale on the x and the y-axis will get you 1 mark. So 1 mark for labeling the axis, the second mark for the scale, and then the third mark is for plotting, and the fourth mark is for joining the plots. You can see at 5.5 degrees Celsius, which is somewhere here, it is 57.5 arbitrary units. So I've got the first dot, and I'm sure everybody can understand this, and I'll just plot it for you all, and then you can see how I've done this. So you can see it was a little difficult. 36.0 is 24 to 45, at 49.5 would be somewhere here. It is 102.5, and that's somewhere here that I plotted it. I'm sorry, it's not very accurate, but then you have to just join these lines, and then you have to join the plots, and that's it. So these were all the points, and now what you have to do is you have to connect them with a ruler, and then you go right through the points. I'm sorry, might not be very exact, and then you correct this down here. So this was the graph that is expected of you. So third mark was for plotting, correct plotting, and then the fifth mark was for joining. Use your graph on figure 5 to determine the rate of reaction when the temperature is 37.5 degrees Celsius. And that's 1 mark. And then explain the shape of the graph between 5.5 and 36.0, and between 36 and 49.5. It says explain the graph, and that is 23 marks completed. So at 37.5, all you have to do was you extend the line and you take it up. I'm not sure I'm getting it very right. And then you read off whatever was there on the rate of reaction on this side. So you just read that off, and that gives you what was the rate of reaction at 37.5, but we want to see this line on the graph. So whatever the figure, whether it's 225 or 222 or whatever, you had to just see this, and you have to read this point of the graph, and this is nearly 225, but I might be slightly off the grid. So please just do it very carefully on your graph, and then you just have to write that you got your mark. What is the explanation? Between 5.5 and 36, enzyme and substrate have more kinetic energy. More successful collisions, more enzyme substrate complexes formed. Between 36 and 46, 49.5, shape of the active site changes, or enzyme denatures, and fewer enzyme substrate complexes formed. 5 and 6, you could see the rate was increasing. Between 5 and it was between 5 and 36 degrees. So between 5 and 36 degrees Celsius, you can see in the graph, this is 36, so this rate is increasing. You can see this graph how it is increasing. That means more kinetic energy, more enzyme substrate complex is formed. ESC is a standard abbreviation. You can use this in the paper. But with 36 and 49, now it's decreasing. So what you had to understand was that enzyme denatures and fewer enzyme substrate complexes formed. So that completes this practical, and I hope this is going to be helpful for you. And all the very best, and thank you very much.